molecular anthropology, which has rather long history. Everything starts with the conception, and the conception was the von Steiner's invention of blood groups in the very beginning of 20th century, exactly at 1900. And it took 19 years for gestation to give birth to the genetic anthropology, to give birth to human population genetics. And the first paper published on this subject was out in Lancet exactly 100 years ago, and in September we will celebrate the centenary of this event. And this paper was about blood groups A and B, and the data were collected in soldiers who participated in the First World War, and actually it was collected in Serbia. And two immunologists, Polish immunologists, Ludwig and Hanka Hirschfeld, published this paper, and since that time we found the age of human population genetics. They found that they considered these two groups as located in different loci, so inherited independently from each other. And this is the summary of their results. And you see here that the A group is more frequent in England, and B group is more frequent in India. So the main inference done was that the human population on the globe have two sources. The first source is so-called European one, Central Europe and Western Europe, and the second one is India and Asia. So it's coming back. Very important is to read the title of this paper: Serological differences between the blood of different races. Now it's forbidden to use this term because it's not politically accepted. And their inference was about the division of humankind to two races. First one with a high rate of A, and the second one with high rate of B. And the mixation took place somewhere between Asia and between Europe. Next one, then much later in 60s, another project also based on blood group was conducted by Watkin, and he used much more donors, much more data. It was published in Heredity journal, and he found the evident climb north to south climb, and he also could identify area with high level of the group A in the area which has a very strange name, Little England beyond Wales. And the main reason of this separation was the influence of farmers from Netherlands. And it was then proved later by using new methods and new markers. The next one was the study conducted by Cavalli-Sforza and Edwards, and you know that Cavalli-Sforza, he is the father of modern population genetics, mainly human population genetics, And this evolutionary tree, is based on 18 alleles of 5 blood groups, and including 15 human populations. And you see that there are distinct separation to 3 global groups: Europeans, Africans, and Asians. And this coincidence of separation, genetic separation, and geographic separation or classification is not astonishing, because genetics usually reflects their geographic proximity. Just briefly, there is the same material, it's the same marker, it's a B group, blood group, also shows the high rate of frequency in Pakistan. The next one is everything was summarized in 1994 in this famous book, which was published in authorship with Benozzi and Piazza, and this is the main or the principal textbook, which is as a Bible for human population geneticists. It resumed all information collected so far in 90s about the serological, about the genetic, about everything, about the polymorphisms in different human populations. This is also the evolutionary tree for human population based on 42 populations, and much more alleles and much more genes. And what is special for this case, that only two loci from HLA, they provide 25% of overall diversity. So, and it was used later to get more precise evolution tree, and based on genetic data. And a new phase started in the end of 80s, when this seminal paper was published in Nature, and it's about the mitochondrial DNA, and it's about the African origin of Homo sapiens. Of course, it reflects only one locus. It reflects only the genetic history. It shows the genetic history of in one side, of maternal side, also called matrilineal genetic history. But it also it was the beginning of genomic era in human population genetics. The counterpart of mitochondrial DNA is Y chromosome, who is inherited only through paternal line. And this publication, this paper of Underhill, which summarized the genetic data of more than 1,000 populations, and which represent the whole diversity on the globe, and using 167 markers, and which fall into more than 100 haplotypes. And here also we can distinguish 3 main ranges. African one is Pakistan with Europe, and with America between Europe and Far East. And so roughly the division, the classification of human population is the same, and it should be the same, based either on blood groups or this novel markers as Y chromosome markers or mitochondrial markers. The next step is the structuring of human genome. This approach provides good possibility to understand the source populations which had contribution to any population in the world. And you see here that the gene pool of different populations, they are described by different colors. And you see here the definite climb from this brown color to these pink colors and these violet colors. And this is the direction from Africa to America. And you see different rate of proportions of ingredients or genetic input of African origin, of European origin, and Asiatic origin, and pure American origin. So we see very smooth change from Africa to America, and it reflects the actual evolution, actual migration of Homo sapiens out in the frame of out of Africa hypothesis, from Africa to about 60,000 years ago, and to America, which was populated, according to recent data, about 50,000 years ago. The next approach is using millions of SNPs or hundred thousands of SNPs to classify the human populations globally. And in this case, we see the same pattern, that the genetic classification fits very well with geographic classification, and you see it's the same pattern, the same classification, almost the same. So we can state that we can even predict any results of genetic study of genetic classification based only on geographic classification. So the spatial classification is the same as roughly, of course, and of course it relates only to the time when the migration was not so intensive as we experience today. Then now about the post-genomic era and genetic maps in post-genomic era, which started in the very beginning of 2000, because the first one was the genetic project started in Estonia. In Estonia, but it was just a collection of DNA samples in general population in patients, but it was not announced as a genome project in terms of mapping, in terms of drawing the atlas of genetic atlas of Estonia. I will be back to this project later, but now I want to show you two projects and two papers published in 2014. The first one is the genetic structure of British populations, and the name of the project is People of British Isles. And the second one is the Dutch project, and you see the figures. I don't want to read it. And these projects are focused not only on demographic history, or it's not pure anthropological, but they also pursue some medical aspects of this mapping. And you see that they included millions of single nucleotide of SNPs, and not only changes but also insertions and deletions. So then, in 2070 and 80, we find much more projects which are focused on mapping, on genetic mapping. And you see here the project Irish DNA Atlas. It was published in 2017. Demographic history of native Mexicans, also in 2017. And whole genome sequencing in South Africa, 17. And this one, no, this one is 17. And which one? And, this one. And the latest one, the most fresh one, is Genomic History of Sardinian Population. All these projects are based on whole genome sequencing. So this is another stage, another level of classification of mapping. And we just, as an example, two maps to show you very briefly, three maps. This one is people of British Isles. You can read that very pronounced structuring of population of British Isles with separation of the Orkney and the same area of small England in Wales. And they used very structured data and very refined data while collecting samples. It's the main issue, it's the main requirement to collect relevant samples. Now we have all possibilities to analyze. Now we have sophisticated programs, sophisticated softs and everything to analyze data. But first of all, we have to ensure the relevant samples, relevant material, raw material for the analysis. And if you put into analysis wrong material, wrong data, not right data, you will get not right, wrong results, shifted results. So the main issue now, and which is the principal one, is the collection of material. And the authors, they stress several times that the material they collected, the material is relevant to the objectives of this project. The next one is Irish, and of course it's a counterpart of the British Isles, and they also found very pronounced structuring of genetic diversity for the population. And I want to stress this thing, that they use only 100, less than 200 Irish individuals with 4 generations of ancestry. So in order to be sure about the geographic origin of the donors, the strong requirement was to recruit or to collect DNA data from individuals with 4 generations, at least 4 generations from the same area. And this is method, it's the same. So and the latest one is a genetic map of Sardinia. You know that Sardinia, it's a unique location in terms of preservation of Neolithic features of Homo sapiens. So far they could preserve the very pronounced traces of Neolithic population, and the population of Sardinia is used as a template for population genetic study, especially when geneticists study the ancient migrations from the Near East to Europe. And Sardinia was one of the crucial points of this migration. Getting back to Estonia, Estonia maybe launched the most, the biggest one project, and they are planning to map or to sequence more than 10% of their population. The population is not big; it's less than 1.5 million of inhabitants in Estonia. But you can read that their objectives is very ambitious, and I'm sure that they will reach all objectives, because they started these projects 18 years ago. 18 years ago, in the frame of another project, and it was conducted by Estonian Biocenter. Yeah, thank you. Now about Armenian genetic atlas. Big difference between the Armenian project and project already mentioned, because we are collecting DNA samples from population which actually geographically doesn't exist. You know that Armenians were forcibly expelled from their native regions, homeland, and but we are going to map, to draw a genetic map, and using DNA samples from the descendants of Armenians who lived here 100 years ago. It's a big challenge, and it's a big difficulty to find out this, to identify these participants, to identify these individuals, and to collect DNA samples. So we are mainly working not in Armenia, but outside Armenia, because Armenia comprised only the small part of refugees 100 years ago, and the majority now are scattered in Near East, in America, in South America, in Canada, and so I thought also that's very difficult to do, because we cannot conduct archaeological, anthropological, and other kinds of survey in Turkey, in Iran, and now in Azerbaijan after the forcible displacement of Armenians from eastern part of Armenia. So, we divided the whole stretch of Armenian highland to 30 regions, and our current activity is this sampling of DNA specimens in order to have the complete coverage of Armenian highland, which was the homeland of Armenian population. And main objective is to reconstruct the demographic history of the region, and because it played crucial role in the migration of population from the Near East to Europe and to Asia. And for the historical purposes, for the national purposes, we want to reproduce the rich spatial mosaic of Armenian gene pool, which originated and flourished during the millennia on the stretch of Armenian highland. And we can address different questions. I don't want to read. And one of them, of course, is the origin, place and time of origin of the Armenian population, which is the question as matter of debate between the historians, and especially between historians who relies only on written documents, on written records. But we are delving deeper. We are dealing with deeper material, with deeper information. And the next one I want also to stress: did Arabic, Turkic, and Mongolian invasions leave traces in modern Armenian gene pool? this question can be answered only using genetic tools, genetic approaches. And DNA sampling, and we use the same approach, collecting DNA samples from individuals whose all ancestors at least derive from the same region, and within about 100 kilometers in vicinity. And so what is the reason of this kind of approach? Using this methodology, we collect a random sample of grandparents' DNA. So we reconstruct virtually the gene pool of our grandparents or great-grandparents who lived on the whole stretch of Armenian highland. Two words about the work of our colleagues. It was published in 3 years ago and based on genome-wide data, and they could, oh excellent, it's enough. They could show that modern Armenian gene pool is a result of mixation of different ethnic groups, of different tribes, which took place, and this mixation took place about 4,000 years ago. Started about 5,000 years ago, and the development, the origin of Armenian gene pool could be dated back to this time. And this is the Bronze Age. Bronze Age, this is the beginning of modern civilization, and the mighty kingdoms appeared that time in the Near East, in Anatolia, and after the collapse of these big kingdoms, the Armenians could preserve their genetic so-called purity, isolation. I don't want to use the term purity, but isolation, genetic or reproductive isolation during at least 4,000 years. This is, and I want to stress that this result was achieved by our colleagues, no Armenians in the list of authors. So, but Armenia, and stressing again that Armenia is very important area in terms of migration, in terms of genetic, as a remnant of Bronze Age era in our region. So Armenian gene pool also, as a Sardinian one, might be used as a template which reflects the genetic construction or genetic makeup of population which lived in this area during Bronze Age. So it fits very well also with the classification of Indo-European language, and you see this is a famous paper of Atkinson, of the group led by Atkinson, and published in Science 7 years ago, and so the good coincidence with the separation of Armenian language from the trunk of Indo-European family, and the separation of Armenian gene pool. About the structure, Armenian gene pool consists mainly two ingredients. The first one is Near Eastern one, and this one, and the second one is Caucasian one, and we have very tiny, very small portion of European influence. So Armenians are mainly indigenous people with tiny ingredients of European origin. Answering the question about the traces of invaders, so virtually no signals of Central Asian invaders in our gene pool, not only in the Armenian but also in Georgia and in Iran. You see the traces of Turkic-speaking populations or tribes only in Turkey and in Iranian Azerbaijan and in Azerbaijan Republic, current modern Azerbaijan Republic. So it was the influence of genetic influence which left traces in these two separate areas, but not in Armenians and Georgians and Iranians. The next one, this is data from our recent paper based on mitochondrial DNA. Excellent, thank you. And we also couldn't find any traces of Central Asian origin in maternal gene pool while analyzing DNA, mitochondrial DNA. And we've found close distances or resemblance between modern Armenians and ancient population, beginning from the Neolithic time and spanning till late medieval time. And our last recent publication based on mitochondrial DNA allowed us to construct a model of genetic continuity between the population of from Neolithic time to modern, and the best model which explains the genetic continuity is this one. So we can state that at least maternal gene pool of Armenians are stable during 8 millennia. Oh. And I want to finalize with this sentence. Please read, just to explain that any project which is focused on medical genetics should rely on population, on demographic history of population, and without this data, without these results, impossible to make unshifted inferences about the evolution of medical traits. Thank you.